Plasmids. Plasmids are small DNA molecules within a cell that are separate from the chromosomes. Like chromosomal DNA, these plasmids are double-stranded, meaning they have two sides of amino acids. Unlike chromosomal DNA, these plasmids are circular, meaning they reconnect on one side. Plasmids can replicate independently within a host, kind of like a virus. But unlike a virus, the DNA is raw or naked, meaning it does not have a film around it. These plasmids can be transmitted from one cell to another. Plasmids in nature are most commonly found in bacteria. These plasmids can be transferred frequently amongst the same type of bacteria or different type of bacteria. This transformation requires direct contact between the cells. This is called conjugation. This frequent transformation between the bacteria allows them to become resistant to antibiotics, and it also allows for rapid mutations. Artificial plasmids are called vectors. These vectors are used by humans to multiply a particular gene. When using vectors, the stranded DNA is removed from the cell and is cleaved using restriction enzymes. The cleaved section is generally the desired trait. On the other hand, you have the vector. That is also cleaved by the restriction enzymes to get rid of that section. These two come together and ligase reforms this recombinant DNA. This recombinant plasmid is then inserted into the cell. This cell naturally divides using mitosis to produce equal strands. There are many applications of plasmids in the world today, such of which is to mass-produce proteins, like to make insulin and antibiotics, to insert a gene for pest resistance into crops, to put a gene that causes bacteria to clean up toxic waste from oil spills. You can create proteins to dissolve blood clots to prevent heart attacks. And when a person's growth is stunted, you can use plasmids to create proteins that increase body growth hormones. The pressing concern in today's world is, has science gone too far? Changing the DNA structure of these organisms. How much is too much? At this point, many of you might be wondering exactly how these plasmids relate to chemistry. I'm going to tell you in just a minute. That's nice. This is an ATP molecule. For all practical purposes, we can make it simpler. By using this. how ATP creates energy is when the bonds break between two phosphate groups, energy is formed. This bond between the phosphate groups is broken because ATP donates a phosphate to the process that the energy is required. So now, the adenosine triphosphate without three phosphates is now adenosine diphosphate. This can be recharged, however, by entering mitochondria. This mitochondria donates an extra phosphate to turn it back into ATP. This process is continued for rechargeable energy. Through the chemical process of ATP, plasmids are able to change the genetic structure of organisms. This process has the potential to benefit humans along with other species in countless ways.